360. All right, will you come up? And, here's the microphone, take it away.
Wants to run the slides.
Hey guys, so my name is Sam.
I'm Grace. I'm Ella.
And we're from ENVS 360. So ENVS 360 was a new course offered last semester about forests. It was taught by Jim Proctor. During this course, we had the opportunity to actually go down to Jim Proctor's forest and in Southern Oregon in Douglas County and kind of talk to some people and do a little case study there. So that's what we're going to be presenting on today, is some of the topics we discussed while we were in Southern Oregon. So the main topics we'll be talking about today are Douglas fir mortality, traditional ecological knowledge, lumber mills, and timber-dependent counties in this situated context. So I'm going to start off by talking about Douglas fir mortality in Southern Oregon. If you look at this picture up here, this is kind of the situation that's happening right now in Southern Oregon. With Douglas fir, a lot of trees are dying, and it's leaving a lot of fuel for forest fires. So a question we asked when talking about this this topic is, how should we evaluate Douglas fir mortality in Southern Oregon? And so just some background on Douglas fir. It's the main dominant species of trees in this area. It grows in low to mid elevation forests in the Klamath ecoregion. This ecoregion is kind of borders, if you look at this map right here, it borders Southern Oregon and Northern California. And you see little darker spots, that's where it's kind of more prevalent, where there's more Douglas fir dying in this area. Douglas fir is an important species in this area. It provides habitat for a lot of different species. It sequesters carbon in this area, and it's also a really important factor for the timber industry, as it has a very high commercial use. So recently, Douglas fir have been dying in this region due to right after drought on water stress sites. And it's making it more vulnerable to other factors, such as the flat-headed fir borer, which is a beetle that will go in when the tree is dying and kill it, leaving a dead tree, and then that leaves a bunch of dead trees in an area, and it makes it a lot easier for forest fires to just sweep through the area and kill all these and just take out all these trees, and that's really, the community is really stressed about that right now, so. Let's skip this. So a way of thinking about Douglas fir mortality is using Mannion's decline spiral. So both of these figures are essentially showing the same thing. It's kind of showing a way to hypothesize Douglas fir death. It has, so all of these factors add up and spiral down, eventually leading into the death of the tree. So these factors, again, include drought, water stress sites, inadequate management practices can be thrown into in there too, causing the species to die off in this area. But moving forward, a better way of hypothesizing Douglas fir mortality is using C.S. Holling's panarchy cycle. So this cycle has 4 stages: exploitation, conservation, release, and reorganization. So the exploitation phase would kind of be, timber companies using Douglas fir, exploiting it, having inadequate management practices, and then that leads into the conservation stage where we are right now, where we're seeing the species die, and we're seeing it kind of react to all this drought and this changing climate, and that will eventually lead into the release of the species and the death of it, which then leads into the reorganization of these forests in Southern Oregon. And that reorganization is going to look something like these forests adapting to drought and more species coming into the forest that are able to withstand drier climate. And then this diagram right here kind of shows how other panarchy cycles influence and impact each other. So say the middle one is Douglas fir mortality, this cycle that is just continuous. And on the top, a large and slower cycle might be something like climate change, so that helps move the other cycles through each other. And a smaller, fast cycle might be like the flat-headed fir borer affecting Douglas fir mortality and causing it to go through the cycle.
Okay, I'm going to be talking about how is traditional ecological knowledge relevant in forest management today, specifically looking at the Cow Creek Bend of Umpqua in southern Oregon. So one working definition of traditional ecological knowledge is knowledge and practices passed from generation to generation, informed by cultural memory, sensitivity to change, and values that include reciprocity. Don't know why the image on the right isn't showing up, but that's an image from the OSU Department of Forestry that represents these core tenets of reciprocity, kind of often in line with a more romanticized and traditional view of TEK that doesn't always account for economic and technological advancements and integrations in indigenous communities. Although this is just one working definition of TEK, and there are evolving perceptions of the term, TEK has largely eroded in practice over the last 100 years, and definitions of TEK have shifted to being seen more dynamic rather than static, which has led to more recognition of TEK in environmental policy. So the Cow Creek Bend of Umpqua is an indigenous tribe located in southern Oregon in Douglas County. In 1854, they ratified a treaty with the U.S. government, which officially recognized them as a landless tribe. And this treaty gave up more than 800 square miles of land for only 2.3 cents per acre. Today, the tribe is working on buying back these ancestral lands with the funds from their endowment, and currently they're co-stewards of tribal lands with the U.S. Forest Service. So what does Cow Creek management look like today in practice? In 2018, the Cow Creek Bend of Umpqua took control of 17,519 acres of land. While these lands were not in control of the Cow Creek tribe, they were heavily logged and replanted as a monoculture of Douglas fir. And the tribal government researched these lands and found that much of this land was historically oak savanna meadows. So this tribe is currently in the process of moving forward with ecological restoration to move these areas back to what they once were in historical conditions. And the tribe uses a lot of, the tribe uses a lot of prescribed burning and fires to reduce fuel loads and ladder fuels and stand density, and just to increase the fire resilience of the forests in managing these forests today. And so we got this really wonderful and unique opportunity to see these lands ourselves in southern Oregon. We toured these lands with Wade Christensen, who is a forester who works for the Cow Creek tribe in southern Oregon. And we saw vast amounts of tribal-owned land recently acquired through the federal settlement, which had been decimated by a recent fire, the Milepost 97 fire. We also saw really diverse forest management styles in practice on our tour, from clear-cut to commercially thinned and untouched forest land. Throughout our tour, Wade really stressed this centrality of tribal forested land managed for a few central goals, including fire management, forest health, and economic profit. So what are some implications of traditional ecological knowledge today in indigenous land management practices? We saw that there are a variety of social, economic, and ecological incentives in southern Oregon indigenous forest management, and thus that this historical romanticized view of TEK that doesn't account for modern economic and technological advances central to the Cow Creek tribe forest management practice is not fully relevant in explaining indigenous forest management today.
So lumber mills are the main party involved in processing logged timber. So the question is how lumber mills can adapt to these changes. So we've noticed a significant decline in lumber mills recently. And this is due to a lot of reasons, but following the timber wars in the 80s and the 90s, environmental culture and regulation had a significant uprise, and this led to supply deficits for timber mills due to a lot of environmental advocacy. So we noticed a lot of closing mills, a lot of increased production per mill, and this contributed to labor shortages, also because young people were less inclined to work in timber management and timber use. Part of this is because of the environmental culture around it, but also the difficulty of the job and the lowness of the wages. And as well as this, automation significantly caused shortages of labor in these rural areas. Also, post-COVID, there was a lot of blue-collar unemployment across the nation, so this all contributed to labor shortages And as well as this, timber methodology was changing a lot. So rather than the commercial thinning, or rather than clear cutting, there was a lot of commercial thinning, which is cutting down the smaller trees to allow the bigger ones to grow and stay big, which is just an example of the conflicting environmental values at the time. As well as this, there's a shortage of lumber from Douglas fir mortality, as we talked about, and also the environmental regulations.
So a way lumber mills are changing is that they're using smaller cuts in diameter of lumber instead of these large cuts. So like Ella just said, they're doing more commercial thinning, and so what that means is that they're cutting down smaller trees, so they're going to be using smaller pieces of lumber in their production. So for example, when we were in Southern Oregon, we had the opportunity to tour D.R. Johnson, which is the main lumber mill down there, and they're usually a mill that produces large decorative beams, but now they're kind of switching more into cross-laminated timber that bands smaller beams together to kind of still make a larger product, but using smaller trees to produce their timber. And this is kind of like the best of both worlds because they're still able to produce the products that they want and make use of it, which usually requires a lot of large timber. And we can see this adapting throughout across a lot of Oregon architectural projects today, where mass timber is building more material that is like more popular and uses a similar concept of using these smaller beams and laminating. And an example of this is the new PDX airport, if anyone has been there. And another way they're kind of adapting to these changes that they're salvaging. They're doing salvage logging on sites that have been burned through forest fires. So they'll request to log a site where it's just a bunch of dead trees, and even though that might have some ecological benefits for the species in that area, it's really important for those timber industries to get profit off of those dead trees.
All right, so as previously mentioned, lumber mills are trying their best to adapt to these issues that are arising. Some, such as D.R. Johnson, are successful and able to stay afloat, but there doesn't really seem to be a one-size-fits-all solution in the timber industry. At the end of the day, there's still a need for timber and a high demand. However, shortages of labor and limited access to timber are the largest obstacles mills in the Pacific Northwest are facing. So moving forward, some questions that we find ourselves asking are, will lumber mills successfully adapt to changing environmental conditions of the forest and new management practices with innovative lumber processing methods? And what will the next 4 years look like with a new president in office, and will it shift environmental regulation?
So these timber-dependent areas, such as Douglas County, it raises a lot of questions around how they'll move forward into the future under the impacts of industrial timber changing and increased environmental regulation. And we got to talk about these issues with Chris Boyce, who's the county commissioner of the area, and he works a lot right at the intersection of policy, both local and federal, economics, and environmentalism, which are all things that are super relevant to these kinds of questions. So he discussed with us the challenges of from Douglas County of the economic transition from being so timber dependent as timber is becoming a less viable option. And a lot of this has to do with Douglas County's history. So Douglas County is built on ONC land, which was land originally given to Oregon and California in 1866 to create a West Coast train system. But due to the ruggedness of the terrain and the value of the timber on the land, it was revested to the Bureau of Land Management in 1937. So income from these lands, 25% goes to the counties, 25% goes to the state of Oregon, and 50% goes to the federal government. So despite a 25% share of the money produced from ONC timber going to the counties, they're still struggling a lot, and a lot of this is because BLM manages for permanent production, so a sustained yield of the forests, and a lot of this is for the environmental factors and the environmentalist values. This contributes to much higher prices from Bureau of Land Management land than from Forest Service land. So despite Douglas County receiving the most funding from 25%, they still struggle. And the question is why they still struggle, and a lot of this has to do with disconnect and misinformation between urban environmentalists who are voting on these issues and the actual experiences and the lived reality of timber industry managers on in these rural areas. So, I mean, I personally experienced a view shift talking to Douglas County Commissioner Chris Boyce and hearing about the problems he faces that urban environmentalists may not consider, because we have these values, but we don't often understand the reality or have empathy around their lived experience. So moving towards having more conversations around local control and the challenges around having more local control, for example, the bureaucracy that makes it very challenging to have effective policy put into place quickly, as well as the purpose of these policies, because there's a lot of conflict between environmentalists who may want old growth forests or timber industry people who rely on the income for their public libraries or their management of the cities. So purpose and speed are both challenges to local control. And in conclusion, Douglas County, we got to talk about a lot of the history and the realities of ONC management and how education would help overcome a lot of these problems. And many other timber-dependent counties are very similar, how overcoming these problems through communication and education to face the nuanced challenges at the intersection of policy, environmental values, and economics will be very important moving forward into the future.
So if there are any burning questions, you can ask right now. Otherwise, there will be a session, a Q&A session where everyone convenes as a panel to ask. Any questions? All right, then thank you so much. So up next is Sadie Meredith Andrews, who's going to talk about the work she's doing with Tamily Weissman, investigating Parkinson's disease, mechanisms of alpha-Synuclein aggregation in zebrafish.
Hello everyone. I'm going to be talking about the research that I've been doing for about the past year in the Weissman lab here at Lewis and Clark, which is entitled Investigating Parkinson's Disease, specifically investigating the mechanisms of alpha-Synuclein aggregation in zebrafish. And so you all may have heard of Parkinson's disease. It's the second most common neurodegenerative disorder behind Alzheimer's, and it is characterized by the loss of dopaminergic neurons, or neurons that produce dopamine, in the substantia nigra of your brain, which is located right here in the midbrain and is very important for motor control and function. And this disease affects about 1% of the population above the age of 60, and because it's neurodegenerative, the symptoms worsen over time. So there are two big categories of symptoms in Parkinson's disease. The motor symptoms include rigid muscles, tremors, and difficulty with movement and balance. And then the non-motor symptoms include memory loss, slow cognitive processing, and behavioral or mood changes over time. And currently there are no known cures to stop the progression of Parkinson's disease. We do have treatments that aid quality of life and help patients with managing their symptoms. One of these is called L-DOPA, and it's the most commonly prescribed medication for Parkinson's disease and increases the amount of dopamine in patients' brains to help manage symptoms. But if we want to develop a treatment that halts the progression of the disease, what we would need to do is first understand the underlying mechanism of the disease and then develop treatments that target that mechanism and hinder it. And so I'm going to tell you what we know so far about the mechanism of the disease and then what we still need to answer to hopefully do that. And so one thing that we know is that Parkinson's disease is sporadic, so it doesn't necessarily have to be connected to a family history. And a lot of times cases are brought about by a combination of genetic and environmental factors. And we also know that the only way to have a clinical definitive clinical diagnosis is through a post-mortem autopsy revealing Lewy bodies. So let's talk about what Lewy bodies are. Lewy bodies are aggregated protein. They aggregate progressively over time and show up in the cytoplasm of neurons. And you have lots of protein in your cells that's doing lots of great things for you to keep you alive and functioning and doing all the things that you love. And you can imagine that if some of this protein got all clumped up and hung out in the cytoplasm of your cells, that might not be super great for your cells. And so that's what we think is happening in Parkinson's disease. And the main protein that is aggregated in these Lewy bodies is called alpha-Synuclein. So let's talk about what alpha-Synuclein is. Alpha-Synuclein in healthy brains is found at the synapse and the nucleus within neurons. And this is what we think it looks like, and it's generally found unfolded in healthy brains. And we know that it seems to be involved in neurotransmitter release at the synapse, so helping neurons communicate with one another, and then also seems to have a role in DNA damage repair in the nucleus. And so let's take a step back and think about why and how we get proteins in our cells in the first place. So we all have DNA, genes that are passed down to us, and these are transcribed into mRNA and then translated into proteins. And these proteins are made up of amino acids, which are building blocks that make up proteins. And the reason they're in the order that they're in is because your DNA says so. And then the order that they're in determines how they fold and therefore what the function of the proteins that result can be. And so one of these proteins that exists in your cells is alpha-Synuclein. And so in this figure, all the letters represent different amino acids. There are 20 to choose from, and they're in this order because the gene that codes for alpha-Synuclein codes for this order of amino acids. And so the big question here is, why does alpha-Synuclein itself aggregate? And we are not completely sure, but we do have some clues. And so I'm going to tell you about the clue that I researched, which is phosphorylation. So phosphorylation is something that can happen to a protein after it's made. And there are a bunch of sites on alpha-Synuclein itself that seem to be implicated in disease. But before we get too far into that, we're going to talk about what phosphorylation is and why it might matter here. So phosphorylation is the addition of a phosphate group to an amino acid along a protein chain. And so here we have an example of an amino acid, which is serine. And then when it's phosphorylated, a phosphate group gets added, and this has a negative charge. And so this might matter for protein structure, because when you add a negative charge to a protein where the amino acids that make it up also sometimes carry charges, this could change the way the protein folds and then therefore the way it interacts. And so for Parkinson's and alpha-Synuclein, when you add a negative charge, it might change the way the alpha-Synuclein folds and therefore might drive it to aggregate. And so another clue that we have for phosphorylation being important in Parkinson's disease specifically is that 4% of the time there's a specific site called serine 129 that is found phosphorylated in healthy brains, so not super often. And then 90% of the time, this same site along the protein is found phosphorylated in disease. And so it seems pretty related to disease. We think something might be going on here. But through previous research in the lab, serine 129 alone does not seem to promote alpha-Synuclein aggregation. And so we looked at these other sites around serine 129 that are also able to be phosphorylated, other amino acids nearby. And the theory is that maybe there's a phosphorylation code to be broken, and so maybe one site, serine 129, being phosphorylated is not enough to drive aggregation, but two sites, one being serine 129 and then one nearby site, could drive aggregation. And so, going along with that theory, that drives my research question, which is, does combined phosphorylation at serine 129 and tyrosine 136 drive alpha-Synuclein aggregation? So, to answer this question, I have to do three main things. The first is I have to have a way to measure aggregation in a system. The second is that I have to have a way to manipulate alpha-Synuclein DNA to mimic or inhibit phosphorylation. And then the third is that I have to express that DNA in a model organism where I can measure potential aggregation. And for that, I use zebrafish. So first, let's talk about how I manipulate this alpha-synuclein DNA. I use DNA plasmids, which are circular pieces of DNA. And there's a lot going on here, so I'm just going to tell you the three most important things that are included. The first is a promoter, which just localizes this protein expression to the nervous system of the zebrafish, which is awesome. And then I also include a human alpha-synuclein gene. And so this is the gene of interest that we're wanting to look at. And this is where I can introduce changes that mimic or inhibit phosphorylation, which we'll talk about in a second. And then I also have to label the expression with green fluorescent protein so that I can see if and where in the zebrafish the protein is being expressed, which will be super important later, and you'll see why. So to mimic or inhibit phosphorylation, what I can do is change the DNA to code for a different amino acid at the site of interest. And so I'm going to use serine 129 as an example, but I would do the same at site 136. And what I can do is change the DNA to inhibit phosphorylation by changing it to code for an alanine, which has a very similar structure to serine but cannot be phosphorylated. And then I could, in another DNA construct, change it to code for an aspartic acid, which mimics phosphorylation because it's also negatively charged, like a phosphate group. And so now that I have these three DNA constructs, one being your wild-type alpha-synuclein, and then one mimicking and one inhibiting phosphorylation at my sites of interest, I have to express this in zebrafish. But first, we're going to talk a little bit about why zebrafish are so cool for this type of research. So one thing is that zebrafish are the only vertebrate that's transparent during development that's commonly used in lab work. And this is great because with that green fluorescent protein, we can look at dynamic processes happening inside of the fish while the fish is still alive, and that's really cool. And then they also have a similar nervous system to humans, which is great for human disease research like Parkinson's research. And as I've kind of alluded to, we can introduce DNA and have the zebrafish express proteins of interest like alpha-synuclein, which is my next point. We can introduce alpha-synuclein. And then also, they do not have their own alpha-synuclein within their cells, which is great because I can attribute my experimental results to the alpha-synuclein that I have manipulated and introduced. And so, to actually introduce the DNA into the zebrafish, first I mate adult zebrafish and collect their embryos. And then I use a super tiny needle with the DNA that I want to introduce inside. And I inject the DNA into the yolk of the zebrafish embryo while it's at a one-cell stage. And then as they grow up in the incubator, for a few days, they'll take up the nutrients in the yolk and also ideally take up the DNA and start expressing it. And then when they do that, they'll look something like this. And so this is a little hard to tell, but the big circle in the middle is a yolk, and then the zebrafish is kind of wrapped around. And all the speckles on the right side are alpha-synuclein that's being expressed in the spinal cord. So that would be a great fish to use for imaging, which is the next step. And so imaging is where I can measure aggregation. So I'm going to tell you how I do that. I use a process called fluorescence recovery after photobleaching. And so here you can see a zebrafish. This one's all stretched out compared to the other one that was in a tight little ball. And you can see that there is alpha-synuclein being expressed in the spinal cord tagged with GFP. And what I would do is zoom into a section below the spinal cord, like the one that's labeled with a red box. And hopefully I would see something like what you see on the left here, which is a motor axon that is labeled. Here it looks gray, but it's the same as the thing above. And you can see lots of different little speckles, and those are presynaptic terminals. And so this is where alpha-synuclein likes to hang out in motor axons of zebrafish. And so I would zoom into a little subsection of the presynaptic terminals and choose one to experiment on. And the way the experiment goes is I take a picture of the terminals before bleaching, and then I would bleach one of the terminals, just use a really high intensity laser, and bleach all of the fluorophores in that section. And then I would take a bunch more pictures every minute after and watch the fluorescence recover. And the important thing to note here is that when fluorescence is recovering, the individual fluorophores that I bleached before will never come back on their own. And so when you're seeing fluorescence recover, it's because new fluorescent molecules from further down the axon are moving into that area, and that's how the fluorescence is recovering. And so because it's all about movement, we can assume that a smaller protein that's less aggregated would move faster through a cell, can move faster in the system, whereas a larger, more aggregated protein would be able to move slower and therefore would recover its fluorescence slower. And so I can do these types of experiments with all of my different constructs and then compare the results to compare the amount of aggregation. And so now that we've zoomed in really, really close to these small changes that I'm making to a protein and looking for effects, we're going to zoom all the way back out to the implications of this for disease research and how important it is. And so for Parkinson's disease in particular, the number of people that are expected to be diagnosed with Parkinson's disease is expected to double by the year 2040. And as we discussed, there is currently no cure. But if we can understand the mechanism underlying the disease, we can develop a cure and help lots and lots of people. Yeah, and these are my acknowledgements. I would like to thank the amazing zebrafish that I get to work with, as well as Tammy for being an amazing mentor always, and Kyle and Willow and Lily for being amazing people to work with, as well as our collaborators and funding sources for the lab. Thank you.
Any burning questions? All right.
Maybe one
Okay, one.
wonderful presentation. How does phosphorylation now factor in the stability? Did you actually get a result with your different phosphomimetics?
I do not have enough data points yet to say for sure.
Okay.
Yes, so I'll keep you updated.
And our last speaker today is Avi Strok, who will be talking about the effects of adult nicotine exposure in Drosophila. Avi is a senior in the BCMB program, working for the past 2 years in Norma Velasquez's lab. And this is work that he presented at the 66th annual Drosophila conference in San Diego. And he loves Drosophila so much that he's going to do a post-bac in the Mitchell lab in the University of Chicago. Starting next year, and I'll let you take it away. But one last thing is he'll be performing at the Fire Arts Show at 8. All right, Avi, here's your mic. Take it away.
You don't need to talk.
Okay. I'm going to try to talk to Mike. Let's see. Can you all hear me just fine if I talk without the mic?
Yes.
Beautiful.
Yes.
How do I make it show up? Awesome. Hi, thank you for that introduction, Al. I'm excited to talk to you all today about my research project investigating the effects of adult nicotine exposure in Drosophila melanogaster. So just to start off, I want to orient us to the public health implications of tobacco use. I'm sure we're all familiar with tobacco. It has this wonderfully addictive chemical called nicotine right here, great at crossing into your blood-brain barrier, activating those dopaminergic neurons that Sadie mentioned, and making you feel pretty good, but it's also great at getting you addicted. And unfortunately, despite the fact that it's addictive, it also is a real killer. Has negative impacts on pretty much every organ system in your body, from your brain to your cardiovascular system, to your heart, to your liver, to your lung, everything. It's not good. It kills about 8 million people a year, and about 1.3 million of these deaths are not even due to direct tobacco exposure, but secondhand smoke exposure. Additionally, out of the 8 billion people on Earth as of last year, there are 1.36 billion tobacco users, and about 80% of these are in low-income countries where resources could be better spent elsewhere. So addiction remains a global health crisis, and we want to understand the genetic basis of nicotine's effects. So to understand that, our lab uses the common fruit fly, Drosophila melanogaster. You might be wondering, Why? Flies are pretty different from us. Despite our many differences, we share a lot of important similarities, especially in our genome. We share about 75% of our disease-related or disease-causing genes, and they're also just really great to work with in the lab. Here you can see a picture with many, many bottles of fruit flies that have been grown. You can grow a lot of them, and we have a bunch of different tests for their both health and their behavioral choices that they can make. So we applied this to nicotine, and in this project, the step that I've been working on is a characterization of nicotine in a control strain of fruit fly. In our lab, this control strain is these white-eyed flies here, also known as white Berlin. And then based on this data that I've collected, we're now working on collecting these same experiments with mutant flies that have single gene differences. In our lab, these are flies that have red eyes. And hopefully using this, if we find any mutants that have differences in their response to nicotine, either a nicotine resistance or a nicotine sensitivity, or differing behavioral preferences in response to nicotine, we can identify genes that are relevant for its effects, map them, and hopefully develop a better mechanistic understanding between how nicotine controls human behavior, and develop potential treatments if we find proteins that we can target. So before we actually go on experimenting on flies, we have to know what equivalent human behaviors we're going to be looking at. We know that nicotine is a real killer for people. It causes increased susceptibility of diseases like cardiovascular disease and stroke, decreased immune response, and so you can be at higher risk for developing pneumonia or bronchitis, and causes an overall shorter lifespan. We also know that nicotine is a stimulant in humans. People take it for that euphoric effect. It causes alertness, an increased heart rate, and like a jittery response in your neuromuscular junction. Lastly, we know that nicotine is associated with behavioral alterations, preference for the drug, increased tolerance, and can also cause changes to our ability to distinguish and prefer different scents. So in flies, what we can do is measure how well they survive in response to nicotine. We can measure how well they climb as a proxy for the stimulant effects. And we can also give them choices between one or another drug that we give them. And so allowing them to determine what they prefer can tell us about these behavioral alterations in response to nicotine. So the overall experimental question for my work is, what are the effects of long-term nicotine exposure in our control strain of fruit fly on their physiology, including survival, locomotion, and egg laying, and also on their behavioral choices, including where they choose to lay their eggs and what they choose to smell, or olfactory choice. So to start off, we have to know exactly how much nicotine we can give the flies before they just die. So in this experiment, we exposed flies to varying concentrations of nicotine. We put it in their food in these vials here at concentrations ranging from 0 nicotine to 1.3 milligrams per milliliter. And then every 5 days, we saw how many flies were alive. So at the beginning of this experiment here, we can see vials full of flies. And then just 30 days later, any vial that was completely dead, we just removed. You can see there's a lot fewer vials. And any of these have between 1 and 5 living flies. So we quantified this data, and here are 12 graphs. This is a lot to look at, but for any one of these given comparisons, what I want you to focus on is the difference between this blue line representing our control flies that got no nicotine exposure, and this red line representing our nicotine-exposed flies. And I hope what you can see is that as time goes on, the probability that any given fly is alive goes down. And at our highest concentrations of nicotine, like 1.3 milligrams per milliliter or even 1.1, we can see that red line drop off way quicker. At our highest concentrations, flies have large reductions in their mean lifespan. But at our lowest concentrations, even though these graphs might seem a little different, we did not see any significant difference in mean lifespan. We saw a little bit of a sex-dependent effect where there was no concentration you can give a female fly without it having a negative effect on their mean lifespan, but these differences were fairly mild. So based on this, we chose 0.9 milligrams per milliliter of nicotine as our basis for long-term exposure in our following experiments, and we exposed them to this concentration for 5 days. This allowed enough time for the flies to really metabolize the nicotine, get it into their system, but not die so much that there's too few to actually test. So the next physiological characteristic that we looked at is fly locomotion. How well can they climb? This exploits a great behavior of flies where if they're in a vial, you tap them all down, they'll slowly climb up as their innate escape response. So after our 5 days of exposure to either no nicotine or 0.9 milligrams per milliliter, we put them all in these little testing apparatuses, We slammed them to the bottom, and every 5 seconds, I measured whether or not they were below this line of acrylic. We counted that as not a successful instance of locomotion, or above this line, and we counted that as a successful instance of fly climbing. We quantified this data, and we can see over time the proportion of flies that are actually climbing increasing to a point and then plateauing. Again, here in this graph, we can see our blue line represents our control, no nicotine-exposed flies, and these flies are climbing a lot more. So we found that nicotine exposure significantly reduces the proportion of flies that are climbing. Anecdotally, I can tell you that these flies exhibit a lot more erratic flight behavior. Sometimes when they're climbing, they'll like zip around, fly, and then fall to the bottom. A lot of them are staying still at the bottom of the vial, just not climbing at all. They're very lethargic. And this is pretty in line with our survival data, where they're definitely dying. But it's a bit confusing given that we expect stimulant effects of nicotine. But it seems that 5 days of exposure is long enough for the poison to be poisonous. So the last physiological behavior that we looked at is egg laying. The number of eggs that a female fly will lay is a pretty good indicator of their overall health and their reproductive health. So after our long-term nicotine exposure, we put flies in one of these bottles for 48 hours in the dark and allowed them the chance to lay their eggs on one of these plates. These plates have a substrate that flies do want to lay their eggs on, and on half of this plate we put in just no nicotine, and on half of it we put on a low concentration of nicotine to determine if they have some behavioral preference or aversion to nicotine. However, for this first part of the experiment, we just looked at the total number of eggs laid on each plate, and as we can see, in response to nicotine exposure, female flies will lay significantly fewer eggs. Without nicotine exposure, a female fly in 48 hours will lay about 12 to 22 eggs, but after 5 days of nicotine exposure, a female fly will lay less than 10 eggs. So we can definitely see this physiological disruption holding true for our egg-laying experiment. However, when we looked at their preferences or aversion to nicotine, what we were able to do is calculate a preference index. Here, anything at 0 indicates equal preference for nicotine and control side of the plate. However, anything below 0 is a preference for the control side, and anything above 0 is a preference for nicotine. As we can see, interestingly, all flies preferred to lay their eggs on nicotine with no significant effect on this preference when we expose them to nicotine. That was a bit odd to see, given that we know that nicotine is poisonous, it's bitter, it should be aversive to flies, but what we know, we have a few hints about what's going on here. We know that female fly egg-laying behavior has something to do with dopamine release in the brain. We know nicotine causes the release of dopamine in the brain. We also know that egg-laying behavior decision-making in females can be driven by low levels of toxic substances. Female flies will choose to lay their eggs on low concentrations of ethanol or vinegar to provide their offspring some level of protection against these substances. That might be playing an effect here, or maybe we just didn't use a high enough concentration of nicotine, but an interesting result nonetheless. The next behavior that we looked at is fly preferences between two scents. So we know that in humans, in response to tobacco smoking, we have decreased ability to distinguish between scents. So after our 5 days of long-term nicotine exposure, we put them in a trap with the option to either irreversibly go into a trap that has a little bit of water in it or a little bit of grape juice that they should be able to smell as it diffuses out of here. So after 24 hours in the dark here, we let the flies choose which trap they go into and quantified it in a preference index once again. Anything above 0 here represents a preference for grape juice. And as we can see, nicotine had no effect on this preference, and all flies had a preference to enter a grape juice trap. This makes sense given that we just gave flies oral exposure to nicotine as opposed to smoking tobacco, and also being able to distinguish where there is grape juice is very, very important for a fruit fly that relies on its sense of scent to determine where food is. So overall, what have we learned about the long-term effects of nicotine exposure in fruit flies? Well, physiologically, I can tell you, it kills them, it makes them climb less, and it makes them lay fewer eggs. Behaviorally, it didn't seem to have any significant effects, but we saw an interesting result where all flies preferred to lay their eggs on nicotine, and all flies preferred to smell grape juice. So where do we go from here? There's a few more behavioral characterizations we could do in our control strain of fruit fly. First, we want to look at learning and memory. We know that nicotine has effects on learning and memory in humans and can alter our risk for developing Parkinson's disease. So that's definitely a relevant behavior we want to look at. Also, temperature sensitivity. How long can a fly make it at high temperatures without passing out? This is a great just general stress resistance measurement, and also could be reflective of a fly's resistance to epilepsy. Additionally, we want to just look at locomotion, but walking instead of climbing to confirm that result. And where we've moved on to now is characterizing these same behaviors in our mutant flies that have single gene disruptions and seeing, can we identify a fly that has a sensitivity or resistance to nicotine? Once we have that, we can use a Splinkerette PCR protocol that some people in our lab have been working on. You might have seen a poster presentation about that earlier today. And we can identify where that gene is being disrupted, and hopefully with that build out our mechanistic understanding behind nicotine's effects. We've done a little bit of work so far. We found one nicotine-sensitive mutant, and we found the gene that was mutated. It's a gene called frizzled. This is a gene that is a receptor in a developmentally conserved pathway called Wnt that we also share. We need to do a little bit more testing so far to determine whether or not this effect is based on the amount of frizzled or just its presence or absence. But we also found, interestingly, a nicotine-resistant mutant. However, this mutant wasn't mutated in a protein coding sequence. This coded for a long non-coding RNA, which will never make it to the final protein stage of translation. But recent research has found out that long non-coding RNAs may play a role in neuronal signaling or gene regulation. So there could be some downstream effects that are causing this relation to nicotine. I'd like to thank a lot of people for their support throughout this project. My wonderful PI, Dr. Norma Velasquez-Oya, who has really helped mentor me and develop my scientific taste throughout this project. Class thesis students in the lab, the other lab members who helped me collect this data, Justin and Lindy for their experimental support, our funding sources, the Rogers Science Program here at Lewis and Clark, the Victoria Affinity Undergraduate Travel Award who helped me present this research at the most recent Fruit Fly Conference in San Diego, and the FOSA organizers for having me come give this talk to you today, and of course, our wonderful model organism, the humble fruit fly. Thank you.
Should I just have everyone come up?
Yeah.
Everyone come up?
Can I just ask one question?
Yeah.
ChatGPT estimates your fruit flies consume about 100 packs per cigarettes per day. Did you calculate your exposure equivalent?
So that's actually a great question. Like, how much nicotine are we actually giving to a fruit fly? Fruit flies eat about 1.5 microliters of food a day. That's about 150% of how much they weigh. But we don't exactly know the correlation between how much they're eating, how much they're getting into their system, how much they're metabolizing, how much they're actually keeping on them, or versus breaking down to get rid of. We've been working on a protocol to actually quantify the amount of nicotine that the flies are metabolizing. That project is still in the earliest stages. We have to first turn the flies into a liquid goop, take all of the protein out of that goop, and then put it in a chromatography machine to separate out all the components, identify the nicotine, and then determine the concentration. But we're still working on the sample preparation step. So great question, but work in progress.
So any questions? Yes.
This is for Avi. How do you distinguish a male versus female fruit fly? Because you have each graph was between male and female.
Yeah. So fruit flies are a lot more sexually dimorphic than humans. They're pretty easily identified under a microscope. Males will have solid black butts and identifiable genitals, and females are typically a lot larger, and they have stripy butts, and sometimes you can see a little egg sticking out of them. That really helps.
Yes. Back there. It's hard to point, right? So you, did you have a question?
Oh, I mean, I could ask you.
Hey.
So like different sets of fruit flies have different behaviors, like how they fight and stuff. Do you think you would ever look at how they fight on what drug with nicotine?
Fight?
Yeah.
That is very interesting. I have not heard of any fighting assays. The most significant interaction between males and females that I've seen us test is the learning and memory assay that we have in the lab. Jess recently got this working where you can take a virgin male fly that's never mated with a female, give it access to a female fly that's already mated before, and after an hour of training, hopefully that female fly should reject the male's advances. That's something that they do. And the goal of that assay is to see, okay, after it's done that, if we expose it to a different female fly at some time later, does it remember that rejection experience and spend less time trying to court them? I can't say for fighting, but at least for the other effing, we can quantify that.
For Sadie, for when you injected the DNA into the yolk, what exactly were you injecting? Like, is it in a medium? Like, where did you get it? How did you make it?
Yes, okay. So, bunch of DNA plasmids, so the circular things, like tons of them in like liquid, and then I dilute that and then mix it with KCl and phenol red, which is to be able to see the solution that I'm injecting. So when I'm injecting it to the yolk, I can tell that I've gotten it where it needs to be. Yeah.
Also, is it naturally fluorescent?
Green fluorescent protein, I'm giving the zebrafish the gene to make a protein that naturally fluoresces green. Yeah.
Oh,
this is a question for Ella. I wanted to know a bit more about your conversations with Chris Boyce, and specifically about the struggles that Douglas County is facing. I know, like you mentioned, a big part of it was they're having to decrease their dependency on like timber sales. But I wanted to hear a bit more about that, and about like the shares and all of that, because I didn't fully catch that.
Yeah, absolutely. So all harvesting on ONC lands, out of all of that, 25% goes to the counties. And the counties are what are supplying the companies, the people, the labor, the resources. And I mean, 25% of the share seems pretty high, but Chris Boyce was talking about how severely underfunded the programs there are. Schools, the libraries, everything there is, I mean, staffing is being cut, programs and resources are being cut, and he attributes almost all of this to increased regulation and sort of environmental awareness. And a lot of that does come from like the urban environmentalists who don't necessarily know the realities of living in a timber-dependent area. And by timber-dependent, like truly it does mean dependent. You know, almost all of their resources come from harvesting. So even what might seem like small cuts to harvesting have huge impacts on the communities and the amount of resources they have access to. Yeah, does that answer your question?
Yeah, I think that does. Thank you so much.
Yeah, I have a quick question for Sadie. Do you have to investigate individually all the other amino acids that are like adjacent to serine 129 that can be phosphorylated, or is it kind of, because you hinted at looking at serine 129 because specifically gets phosphorylated in diseased neurons. Is there other hints about that, or is there like a limit to what counts as adjacent that you would want to have to run tests for each of them individually?
Yeah, so only a subset of like the total amino acids that exist can be phosphorylated, so there's that like limiting. And then we have like 3 surrounding ones in the lab that we look at because they're about like 4 amino acids away-ish. But also, I had another thought talking about that I lost. But yes, nearby-ish, and also able to be phosphorylated. Oh yeah, and then like also all of these other nearby ones, I think, have like some previous research, somewhat connecting them to the disease as well in some way or another, or other people are looking at them as well.
I think we have time for one more question.
First of all, you all did an amazing job. Good job in your presentations. I'm a little biased towards Sadie's, but I'm actually a question for Avi. So, 2 simple questions. One, I hope it's simple. like 5 days of exposure for a little fruit fly. What does that convert to for human years, right? So percentage of their lifetime, because that seems like a short exposure, but I'm assuming it's actually quite long.
Yeah.
And then the second question I have is, it looked like on one of your graphs that a low concentration might actually increase survival. So I wonder if you could comment on that.
I can speak on both of those. So I don't know. So this is not citing any specific data. This is kind of a folksy knowledge I picked up when I attended a fruit fly conference. But what I heard is that 1 day of an adult fly's life is about equivalent to 1 year of human lifespan. In a laboratory setting, a fruit fly can live like about a month. In the wild, they live for about a week with all the environmental. They're not living the good life of just getting to sit in a vial and eat food and poop all day. So I think it's equivalent to about 5 years of nicotine exposure in humans. And as for what the data looks like at low concentrations, whenever I quantify this data, I'm seeing that while I do see kind of aggregation of the lowest concentrations, 0, 0.3, and 0.5, having similar survival rates as compared to the middle and the higher ranges of concentrations. For the first 15 days of the experiment, the relative mortality rates and risk to death seem to kind of flip-flop all over the place before sort of stabilizing at the later dates. Interestingly, when I quantified it, even though the mean survival time wasn't different, these lower concentrations did have increased survival at the 25% mortality. They lived longer. So it seems that on the short scale, it might have some sort of protection. Maybe this is a sample size issue. It's hard to say, but that is a real observation that you were observing. Yeah.
Thank you all for excellent presentations. And if you have any questions, you can probably come up and chat with any of the panelists.
Thank you.
Thank you so much. Really wonderful, interesting thoughts. Good job.